Good day, everyone. Today we will be discussing the Krebs cycle, also known as the citric acid cycle or TCA cycle. So, first and foremost, I will be discussing the first half of the Krebs cycle. And I'm Peachy Javier, your reporter for this part. So, this process takes place inside the matrix of the mitochondria, known as the powerhouse of the cell. And its main job is to break down acetyl-CoA, which comes from the food we eat, and slowly capture energy that our body can use. It works like a repeating circle, and every turn helps produce energy for our cells. Now let's begin with the first two reactions. So for the first step is the formation of citrate. We start when a 2-carbon molecule called acetyl-CoA combines with the 4-carbon molecule called oxaloacetate. And with the help of the enzyme citrate synthase, they join together to form citrate, which has 6 carbon atoms. And this is how the cycle officially starts. For the step 2, the isomerization to isocitrate. So citrate rearranges its structure slightly. Using what enzyme? Aconitase. It loses then regains a water molecule, turning into isocitrate. So this small change gets the molecule ready to release energy in the next steps. Now moving to the next two important reaction, which is the first and second oxidative decarboxylation. So the third step is the first oxidative decarboxylation, wherein the isocitrate is now ready to release energy. And with the enzyme isocitrate dehydrogenase, it loses 1 carbon atom in the form of carbon dioxide. And this is the gas we breathe out. At the same time, it converts NAD into NADH, a molecule that carries stored energy. The new molecule is now formed alpha-ketoglutarate. And for the fourth step is the second oxidative decarboxylation, wherein alpha-ketoglutarate undergoes another change. And it helped by the alpha-ketoglutarate dehydrogenase complex, wherein it loses a second carbon as carbon dioxide and produces another NADH. So it becomes succinyl-CoA, which holds a high energy chemical bond. And to summarize my part for the first half of the Krebs cycle is first, we have released 2 carbon dioxide molecules and formed 2 NADH that will be used later on to make more energy.
So good morning everyone, this is the second half of the Krebs cycle. I'm Ronamae and for step 5 to 6, I will continue with the second half of the cycle, starting from the succinyl-CoA. And for step 5, this is the formation of succinate and ATP. So the succinyl-CoA still contains a lot of stored energy. With the enzyme succinyl-CoA synthetase, that energy is released and used to make GTP, which is equal to 1 ATP. The direct energy of our cells use immediately. During this reaction, succinyl-CoA also turns into succinate. And then for step 6, this is the oxidation to fumarate. So next is succinate is processed by the enzyme succinate dehydrogenase. This reaction removes hydrogen atoms and converts FAD into FADH, which is another type of energy carrier. Now the molecule now becomes fumarate, getting closer to completing the cycle. So for step 7, now we complete the cycle so it can repeat again. So fumarate then adds 1 molecule of water with the help of the enzyme fumarase, and removes and transforms into malate. So this is just a structural adjustment to prepare for the final step. Then for step 8, finally, malate is oxidized by the enzyme malate dehydrogenase. This reaction produces one last NADH and changes malate back into oxaloacetate, the exact same molecule we started with the beginning. Now it is ready to combine with a new acetyl CoA and start the whole cycle all over again. To to wrap up, here's what one full turn of Krebs cycle produces. So this 1 ATP or GTP, this is direct usable energy, 3 NADH high energy carriers, and then 1 FADH another energy carrier, and 2 CO2 is released as a waste gas. So the NADH and FADH will later go to electron transport chain where they help produce much more ATP. So this makes the Krebs cycle very important because it effectively turns the nutrients from our food into the energy our body needs to function, grow, and stay alive. Thank you.